Hi, everybody. This is the review for the final. It'll be a little bit more cursory than before because you have 2 hours to work on this final. And given that there's that abundance of time, I think the review can be a little less detailed. So we start off looking at the kidney and the renal system, and you certainly have to know that significantly both the structural and functional unit is something we refer to as the nephron. It consists of the glomerulus and then the duct, effectively the tubular system that emanates from it. We look at the significance of it with regard to why it's so interesting. We have two different types. You have the juxtaglomerular, the juxtamedullary and the cortical nephrons. The cortical ones are certainly designed for most of the solute resorption and secretion and processing of the various solutes in the filtrate, either adding to or removing, bringing them back into the circulation, versus water concentration, which is all about the juxtamedullary nephrons. Significant, the cortical nephrons have the peritubular capillaries versus the vasa recta. So important for urine concentration and that whole countercurrent system that is there. We realize the unique nature of the blood supply because you have arterial and afferent, or incoming arterial and efferent arterial, and the fact that physiologically, normally, an arterial is going to lead, that leads into a capillary bed, is going to exit as a venule, having given up its oxygen and nutrients. Not so here, because the significant driving force is all about effectively creating the filtrate, and rather than have to rely on an energetics, filtration is a passive process as long as there's sufficient in this case, not a concentration gradient, but a pressure gradient. So I think that's a significant feature as well. The kidneys are great because they can control flow based on pressure and chemical sensors. So they have a series of mechanical sensors or pressure sensors, and a series of basically solute or salt sensors that principally control flow in and out of the glomerular apparatus by way of the afferent arteriolar structure. And so I think that's really significant. The proximal convoluted tubules are 2/3 of all the absorption and secretion occurs there, perhaps more than any other area. The distal convoluted tubules, much more secretion, a little reabsorption, and again, the processes that are in the loops are somewhat unique. We do see a little bit more solute movement in the cortical nephrons, but in the juxtamedullary, we have that countercurrent system with water going out in the descending limb and solutes being actively transported in the ascending limb. That's there. A unique structure in all of these nephrons is the juxtaglomerular apparatus, which is so really important with regard to balancing that renin-angiotensin-aldosterone mechanism. That particular mechanism is fascinating in that, again, it relies on a series of pressure and solute or salt sensors that are able to determine whether or not blood pressure falls, and if so, then those juxtaglomerular cells will effectively send signals that will deal with those subtle modifications in typically we find them in either the ascending limb or in the convoluted tubule, and secrete the renin, which is going to act as a catalyst and then catalyze going forward the formation of angiotensin from its precursor form, which is in the bloodstream, to angiotensin 1, and then in the lung, angiotensin 2. So that group of cells that is so specialized is called the macula densa. They produce that's where the renin granules are located. The signaling mechanisms are there in the juxtaglomerular apparatus. You have those mesangial cells in between that deal with it. Filtration is an interesting process in the sense that we filter approximately all of the blood 2.5 times an hour, generating about 160 to 180 liters of filtrate, of which about 99% of it is reclaimed. So the key issue is the high pressure filtration system that's so significant with regard particularly in and around the the pressures that are associated, but particularly the pressures entering into the glomerulus. More so, I would think in the afferent arterial, probably than anywhere else, is really what's going to dictate that process. We know what the role, you should know all the roles of what angiotensin, once it's activated, does. There's 4 different things. What it does is it basically, as its name would suggest, tenses blood vessels, tightens them, increasing blood pressure that way, like a sympathetic nervous system would do. Increases your thirst, increases the release of antidiuretic hormone, and activates aldosterone, which brings in salt, and then water follows. So I think that's really significant. The sympathetic nervous system is a major controlling blood flow into the kidney more than anything else. So again, normally the kidneys function, the sympathetic nervous system takes over, and basically in fight or flight, then we really limit the blood flow that going into the kidney, therefore the amount of activity that's done there. So I think that's all important stuff to remember. Again, the countercurrent multiplier and countercurrent exchanger are really the way that urine is concentrated. Okay, the multiplier does the concentration. The exchanger continually removes those solutes from that area, so the gradient is maintained, that osmolar gradient that is maintained. The convoluted tubules utilize mostly an active transport mechanism, more so than anything else. There's a certain amount of diffusion, and perhaps more so than that, the primary active transport, we see a lot of antiport and co-transport associated with secondary active transport. So I think active transport certainly predominates. The critical way of evaluating the pressure has to do with what is known as that mean arterial pressure. And you recall in blood vessel unit, we were able to basically generate that number by looking at the difference between the systolic and diastolic, dividing by a third, and then adding that to the diastolic. So as long as that, in the book would tell you between 80 and 180, but I think it's the low side is undoubtedly accurate. The high side might be a little bit high. So maybe 80 to 160 or 150, because that, if your mean arterial pressure is 150, you're in trouble. Okay. And I really think that that'll disable the mechanism itself, in my own opinion. And I think a lot of textbooks would bear that out. Part of the important process is that transport maximum to recall. That's the idea of making sure that we don't excrete it. When we are excreting solutes that should be reclaimed, whether they're amino acids, whether they're glucose, anything along those lines, they should not appear in the urine normally. Only if there are excesses, and the excesses generally mean that there's been an exceeding of something called the transport maximum. We went into that in quite a bit of detail. So the glucose those transporters that involve the glucose are really, the glucose kind of is co-transported along with sodium, mostly in the proximal convoluted tubule. And I think those are important. You should know about the urine, roughly the specific gravity, what the components are, that it shouldn't have any cellular elements. It should have basically just have the pigmentation associated with basically the urobilinogen, which is really an offshoot of heme. That's there. The acidicness, the somewhat salty characteristic that's associated with it, a variety of agents that are found in there. And you can look in detail what those are that are all part of it. Now, as we look more into reproductive physiology, and there'll be some more questions dealing, of course, with renal as well, as we look at reproductive physiology, and again, some of these things, so I think you need to know about the role of things, the testes and the ovaries, the duct system, what their role is, whether it's producing sperm and eggs, testosterone, estrogen, or progesterone, whether it's like in the epididymis, where sperm are basically mature and to some extent are activated. This certainly the role of the glandular functions, the seminal fluid, really providing a lot of the nutritional elements that are there, the volume, a variety of chemicals that are necessary, the activation elements. The prostatic fluid seems to serve a role of activation, as well as imparting that sort of milky, creamy consistency. that we associate with the ejaculant. I think you need to be familiar with testicular cancer, certainly the relationship between the undescended testicle, that's part of it, that's there. And you remember that the fancy term for that is called cryptorchism. We know that the descent of the testicles normally found in the abdomen, really in an analogous area to where the ovaries are, and they're descending along with the other structures that are associated with the spermatic cord, the lymphatics, the blood vessels, the nerves, the ductus deferens. That is all to create that temperature gradient that's so important for the usual development. The fact that there's a relatively elaborate group of blood vessels, that pampliform plexus, that absorbs the heat efficiently as well. And also the fact that that's a very unique blood supply system is that there's no communication with the sperm per se, because they would be antigenically foreign, having been produced long after birth. Not being started, we don't get sperm production until, which is different than the egg, obviously. We don't get sperm production until adolescence. So understand that where the components are, mostly seminal fluid, then prostatic fluid, the bulbourethral glands, maybe a little bit of lubricant. That's really not so much part of the fluid, but really serves more of that lubricating function. So it can pass the rather generous amount of sperm, probably an average of about 300 million per ejaculation. And that's typically about the numbers. The prostatic cancer, the guidelines that we utilize for that, PSA, digital rectal exam, what controls, you should recall what controls erection and climax, whether it's in the male or the female, the relationship, which one's para and which one's sympathetic. Know the roles of those hormones, everything from that whole axis, from the GnRH, remember high levels or low levels of either testosterone or estrogen, their impact as they're detected, along with inhibin as they're detective, and whether or not they stimulate the release of GnRH from the posterior pituitary, which then signals the release of LH and FSH from the anterior pituitary, and a specific role. Luteinizing hormone is about producing estrogen, testosterone, and progesterone, where follicle stimulating hormone is just that, it's about the follicle. So I think that's probably a very significant feature. The other things, and I'd certainly a little bit about what nitric oxide does vis-a-vis the erectile process, how it works with regard from those things, really the constituents, what makes those things up. We know that all of those sex hormones are cholesterol based, they're fat soluble in their nature. The different things that facilitate the traversing of the egg. Remember, the egg is, unlike the sperm, is effectively ovulated out into the abdominal peritoneal cavity and facilitated by a variety of means. You should be familiar with those means that are there. Understand some of the things that can go wrong, particularly with regard to when we're looking at sexually transmitted diseases. What happens with what is pelvic inflammatory disease? What's the significance and how it relates to ectopic pregnancies, or with regard to early hysterectomies or infertility? How do we avoid it? What's the testing that we use for cervical cancer? What's its association with human papillomavirus? Understanding the distinctions between the menstrual and ovarian cycles, particularly how they relate to one another. What are the primary hormones? When is estrogen most active? When is progesterone most active? When is FSH most active? When is LH most active? So all those, as you recall, that sort of detailed chart that have that graph on one of the slides that demonstrates those four things, as well as endometrial development, the role of the spiral arteries, the spasms, what's the sequence of events with regard to that, once that effectively trigger and result in menstruation, the distinction between the functionalis and the basalis, and the spiral arteries and the radial arteries, and what they're all about. And of course, the relationship as to why breast cancer spreads and the makeup of the breast. And there isn't a lot of physiology so much other than the glandular structure that begins to develop at or around pregnancy, and certainly after birth in preparation for lactation. I think that's important. So where are the activity, those types of things? What happens when the system, like with aging? What are some of the problems when post-menopause or in early menopause when the ovaries are dysfunctional? What happens with regard to estrogen? What are the adverse effects? It's things like osteoporosis, things like increased risk of breast cancer, increased risk of blood clots. What happens with all of those things? So the events that are associated with menopause and the events that are associated with the start of menstruation with menarche, what triggers that? The role that inhibin plays. Where is there some logic as to why all of a sudden that occurs at a specific point in time? Okay, so I think those are all part of it. Identifying what parts of the cycle are which, the ovarian cycle, so obviously the menstrual cycle with the developmental portion of the cycle and the glandular portion of the cycle, the ovarian cycle. So remember the names that are associated with all of those things that are there. Where does fertilization occur? The role of the corpus luteum and the corpus albicans are also important. A little bit about STDs. Certainly, as you remember, the ones that are most commonly found now, problems that are associated, and we did touch on that as part of the lecture material. That is there as well. So I think all of that is worth recalling. And then when we look at the developmental area, and again, this is still somewhat preliminary as I've not completely finished all of these questions from any of these areas, but this is just sort of the generic kind of overview. That's part of it. What is capacitation? What is the role of the acrosome? Where do we find the genetic material in the sperm? How is that eventually released? What product is sort of removed from that acrosomal helmet area that covers it to allow the penetration, as it were, into the egg itself? Look at the distinction. What's a morula? What's a blastocyst? What's a cytotrophoblast? Those things that we discussed toward the end that last week. Why is HCG so important? And also the derivation. What comes from ectoderm? What comes from endoderm? What comes from mesoderm? What are in those layers that are there? It's very distinct. It's all either in the PowerPoint or the lecture notes, and I think you should be around and look at it. When do we see the transitional points between embryo to fetus? I think that's also very relevant. The structures that are associated with the fetal heart, I think that's a really significant point of emphasis, whether it's the foramen ovale or the ductus arteriosus. What do they actually what are they accomplishing with regard to the fetal blood flow and the distinction of what's flowing in by way of the fetal arteries, what's flowing out by way of the fetal arteries, what's flowing in by the role of the fetal vein. Okay. Understand what goes on at birth with regard to active and passive immunity. I think that's important. Also, certainly know about thalidomide and why that was so significant. That is there, as well as fetal alcohol syndrome, the role of smoking. And if I didn't emphasize it, one thing we see associated with smoking is a real diminishing of the availability of oxygen to the fetus and to the embryo and the fetus as it develops. And that can be really very, very significant. Some of the things like rubella and toxoplasmosis are all significant. A little bit about cytomegalovirus is also significant. And then as we get on to the flip side of all of these things, as we get toward aging, what happens associated with the skin? We went over respiratory diseases. You should review them and see some of the things that are part of it as well. What happens in the nervous system? What are some of the hallmark things like the plaques and tangles and chromatic material deposited vis-a-vis Alzheimer's? When does the male usually start to suffer with sexual dysfunctions and infertility? What are cataracts? What goes on with the digestive system? So there's a lot of this material. And so these are, I think the questions are to be somewhat generic. I just think you have to review the material. And I think since you have such an ample amount of time, I think you'll be able to find these answers. So thank you very much. That concludes this comparatively brief review compared to the other ones because it's a different format. And if you have any questions, just get in touch with me. I will be sort of in and out on Monday if you're on campus on Monday, and make sure that you dropped off any of those exams that I have to get. And I'll be sending an email about how we're doing the exam on Tuesday.